Hello, everyone. My name is Kevin Beatson,
My name is Nada Ahmed.
and thank you very much to the BJS Academy and the European Society of Endocrine Surgeons for giving us the opportunity to present our group's work on circulating extracellular vesicles as diagnostic markers in indeterminate thyroid nodules. This work was presented at the 10th Biennial Congress of the European Society of Endocrine Surgeons in May of 2024, and we're very grateful to have been awarded the BJS Prize. We have no conflicts of interest to declare. Thyroid cancer has been increasing in incidence over the last 4 decades, not least in part due to improved imaging techniques and diagnostic methods. Most recent global cancer statistics are in line with this, with thyroid cancer ranking 9th highest in incidence from 2020 to 7th highest incidence of 2022. It is now the 5th highest incidence cancer in women. Thyroid nodules are prevalent and can be palpable in up to 6% of people in the Western world. Evaluation of a thyroid nodule includes determining the thyroid status of the patient and whether there are any local symptoms that necessitate treatment. However, the interest of our research is in the assessment of whether these are benign or malignant. Patients will typically undergo ultrasound assessment, and any suspicious features will require fine needle aspiration. In the UK, ultrasounds are graded from U1 to U5, but elsewhere in the world, they will use other scoring systems such as TIRADS. Fine needle aspiration will result in outcomes ranging from inadequate sampling to benign to definite cancers. However, 20% of these fine needles aspiration cytology results will be indeterminate. Of interest to our project is what is classified as Thy3F nodules, or Thy3F cytology. Elsewhere in the world might be scored as Bethesda 4. These cytological samples suggest a 15 to 30% risk of malignancy, and most patients will typically undergo a diagnostic hemithyroidectomy, exposing the patient to risks of surgery. Liquid biopsy is a concept that's been gaining traction over the last few years. This involves the sampling of bodily fluids such as blood or urine to diagnose or assess a cancer in the body. Some constituents of bodily fluids of interest include circulating tumor cells, DNA, and RNA. The advantages of liquid biopsy include non-invasive sampling, non-invasive diagnostic method, a rapid test, and also liquid biopsies also permit real-time assessment and allow repeat measurement of a cancer. One constituent of bodily fluids that is of interest in recent years as liquid biopsy biomarkers are extracellular vesicles. These are phospholipid bilayer enclosed nanoparticles that are anucleate and cannot replicate. There are 3 broad types based on their biogenesis and their size. Of interest for us are the larger extracellular vesicles known as microvesicles. Microvesicles are formed by direct budding of their parent cell, whether it's a healthy cell or cancer cell. They range in size from 100 nanometers to 1,000 nanometers and can carry signatures from the parent cell, including surface proteins and bioactive cargo, such as, again, proteins or microRNAs. For the purposes of this presentation, we will continue to refer to microvesicles as EVs.
Patients with Thy3F nodules were recruited from University College London Hospital. After written informed consent, a blood sample was drawn right before diagnostic hemithyroidectomy, before the induction of general anesthesia. Then, patient samples were assigned to benign or malignant groups according to final histopathology. And healthy volunteers were also recruited from the hospital and from the university. Healthy volunteers and patient groups were all matched for age, gender, and body mass index. And also, within the patient groups, patients with Thy3F benign and malignant Thy3F nodules were also matched for nodule size to minimize the effect of confounding factors. First, we sought to measure the plasma EV concentrations and whether that can help us differentiate between the patient groups. And that was done by flow cytometry. Flow cytometry is a high-throughput single particle analysis technology where a laser is shown through the sample, particle by particle, And according to how the particles scatter light, we can get information about particle size and also can excite any fluorescently labeled markers on the surface of those particles. After obtaining the blood samples, EVs were obtained by differential centrifugation, first to get rid of the blood cells and then to eliminate the platelets, ending up with platelet-poor plasma. Then EVs were pelleted and then incubated and stained with fluorescently labeled antibodies, and then analyzed on the flow cytometer. For us to confirm EV isolation, we were able to visualize EVs by transmission electron microscopy. This is an example of our EV gating strategy, where any particle falling within the EV size range, which is less than 1,000 nanometers, and expressing phosphatidylserine, which is a phospholipid that is expressed on the surface of EVs and is, in this project, we have used it as a generic EV marker. As you can see, the total plasma EV counts or concentrations were significantly higher in patients with 5-3F nodules, both benign and cancer, compared to healthy controls, with a high area under the ROC curve of 0.945. Out of the total EVs, we've measured the concentrations of subpopulations of EVs expressing other surface markers, including the cancer markers CXCR7, which is the atypical chemokine receptor 7, CD147, and Syndicin-4, as well as the epithelial cell adhesion molecule, representing an epithelial marker. And as you can see, all of them were significantly higher in patients with 5-3F nodules, both cancer and benign, compared to healthy controls. But there was no significant differences between their concentrations between benign and malignant 5-3F nodule groups. Total EVs and EV subpopulations expressing these markers were able to differentiate between patients with 5-3F nodules compared to healthy controls, but that can be done by an ultrasound. Therefore, we postulated that other EV markers can help us differentiate between benign and malignant 5-3F nodules. To do so, we've investigated the protein profile of EVs, and that was done using proteomics, specifically liquid chromatography mass spectrometry. To do so, EVs were isolated based on their size by size exclusion chromatography, which yields a purer EV preparation with minimal plasma protein and lipoprotein contamination. Then the EV proteins were digested into their constituent amino acids and peptides and ran on the mass spectrometry machine, specifically timsTOF. And according to the mass-to-charge ratio, we can identify which amino acids and peptides were in the sample, and working backwards by aligning the data to the human proteome, we can identify which proteins and their quantity that was originally in our EV sample. A total of 1,034 proteins were identified across all the samples. 17 proteins were exclusive to cancer, while 36 proteins were exclusive to benign group. 3 EV proteins were significantly upregulated in patients with malignant 5-3F nodules compared to benign, while 20 proteins were significantly downregulated. Out of those, the 3 significantly upregulated proteins, kallikrein 11, A1AG2, which is orozomucoid 2, and the small integral membrane protein 1, were all identified in previous literature to be significantly upregulated in thyroid cancer tissues as well as cell lines. And they were also identified as upregulated in other cancers. In the third part of the project, other than proteins, we've sought to identify other EV cargo, specifically microRNA. MicroRNAs are small non-coding RNA molecules that are around 25 nucleotides in length. They are mainly inhibitory in function, and they bind to the messenger RNAs temporarily or permanently, preventing them from being translated to their proteins. We use the commercially available kit, the ExoRNase kit from QIAGEN, to first isolate the EVs from the plasma samples and then extract RNA from them for further microRNA next-generation sequencing. A total of 650 microRNAs were identified in the EVs across all samples. 120 microRNAs were exclusive to cancer group, while 90 were exclusive to the benign group. A total of 5 microRNAs were significantly differentially expressed between patients with malignant compared to benign Thy3F nodules. The significantly upregulated microRNA shown in the top row has been previously shown to be significantly upregulated also in serum from well-differentiated thyroid cancer patients compared to follicular adenoma patients. 2 of the significantly downregulated microRNAs were actually shown to be tumor suppressors for thyroid cancer. Interestingly, a novel microRNA has been identified in our EV samples and was only expressed in patients with the borderline tumor, non-invasive follicular thyroid tumor with papillary-like nuclear features, or NIFT-Ps. After identifying those candidate protein and microRNA EV biomarkers, we're now undertaking a validation of those candidate markers on a bigger patient cohort, where microRNAs are being validated by polymerase chain reaction, or PCR, and the protein markers are being validated by Western blotting, ELISA, or flow cytometry. We have also started a collaboration with AI experts to see whether machine learning can help us identify other potential EV biomarkers. And we're aiming to bring back flow cytometry to develop a diagnostic assay in combination with those other techniques. Thank you very much for listening to our presentation, and we would like to thank the BJS Academy and all our supervisors and collaborators and funders as well. Thank you very much.
Thank you.